Hello and welcome, Neil Dougan here. We're looking at Module 14 Science today. We're looking at the chemical basis for cellular functioning. So we're looking at the science of it. Now, clearly we don't have in a short video the opportunity of giving you several university degrees worth of knowledge about all these chemical processes. We're absolutely no intention of talking about all those things that you would study in that type of context. However, there are some basic broad brush areas that I want to cover off. So firstly, I just want to talk about the first principles issues of it, and one of them is that compared with the type of experiments that you might do in high school in a flask or something like that, or in a dish, which are very, very simple. So like we're talking about extremely simple chemistry, like the chemicals themselves are very simple. The chemistry that's going on inside our body, inside our cells, is really complex. It's really, really complex. So I just want to just point that out to you, that this chemistry that we're actually going into here involves enormously complicated bits of chemistry. This type of chemistry that we're talking about here is nearly always mediated. So we're forming from a bunch of reagents, some other chemistry, so they come together and they do stuff. In amongst all of this, there's some form of mediation in nearly all cases. So the mediation will be generally some type of, more often than not, will be an enzyme involved in it. So this is a specific protein that's been manufactured for the purpose. And what I just really want to make clear at this point here is that we're dealing with a situation where we've got a whole lot of complex chemicals already, and for them to get together and do whatever it is that they need to do, they need another very, very complex piece of chemistry to come in and manage that process. So it's either going to speed it up or slow it down or just make it even possible for the chemical reactions to occur. These mediating chemicals are the topic that we're following when trim here. So we're focusing on these chemicals that come in and either hurry it up or slow it down or make it go in huge volumes or only in very tiny volumes. The next aspect of it that I wanted to talk about is that nearly every one of these chemical reactions that we're talking about is what they term being up gradient. And what that means is that we've got these bunches of chemicals here ready, and they're ready to join up and do whatever they're going to do here and turn into these other chemicals over here. For that to happen, not only do you need to have this mediation process going on, but under nearly all circumstances you need to actually have energy supplied into them. So these chemical reactions are nearly always chemically they're energy demanding, so they actually need energy to be introduced into them. Now that energy is introduced generally through other chemical processes, but this is not the normal natural laws of that we talk about in physics. So we talk about the idea of entropy in physics, where generally everything's trying to devolve its way down to lesser and lesser structures and lesser sort of setting, so that the energy is actually given off all of the time. So what we're talking about here is something which is against this sort of principle in physics, and it requires a deliberate decision to actually introduce energy into the system so that all of this complex chemistry can occur. So I mentioned in yesterday's video that nearly all of these chemical reactions that we're really focusing on, so like once again we're only carving off a very small piece of the chemistry that's going on inside the cells, but it's the part of the chemistry that is really useful and interesting to us. But inside of this piece of this bundle of chemistry, it's nearly all mediated by enzymes, and enzymes are proteins, and proteins are made inside the cell from copies of part of the DNA. So the processes of the DNA instruction set and how it goes to make these proteins is once again, it's out of the scope of what we're talking about here, but in brief, a very small section of the DNA is copied into a thing called an RNA, piece of RNA, and that piece of RNA then goes and gets copied into a protein. In this case we're talking about enzymes. Now, there's all kinds of things that come in here to have an influence on the creation of these enzymes. And one of the things you may well have heard people talk about, it's sort of talked about in the common domain nowadays, is a thing called methyl markers, and these are bits of chemicals which come in and bind onto the backbone of either the RNA or the DNA. So it can actually they can bind onto the DNA or onto the copy of it, which is the RNA. When these methyl markers bind on, they completely, or they have the potential to completely cripple or change the instruction set. So in other words, they modify the instruction set. So we've got this perfect DNA giving out all this information, and then we've got this mechanism where something can come in and put a stop or put a change or cruel the functioning of the manufacturing of these enzymes. So methyl markers are just one of many different ways in which these epigenetic forces, so these are the forces from outside of the strict sense of the blueprint, the genetic blueprint, come in. However, we're not going to go into any of the other ones. Just take it as understood, if you wish, that there are a great many different ways in which this whole chemistry can actually be interfered with. So I promised I'll keep this short, so I will. There's three main categories of the chemistry that I want to just have a bit of a focus on, and the first one is what they call active transport. So active transport refers to the process of actually moving the component material or the RNA itself or other sorts of instruction sets actually around the cells and from one cell to another. And this is something you may not be aware of, but most cells in the body actually have little microtubules which come off the side. So if this is a cell, the little microtubules will come off, and there might be another cell over here, and that microtubule will attach to it. And these messages and this information can actually be passed down all these microtubules. And the research at the moment is talking about cells having a reach from these microtubules of as many as 100 cells. The level of communication that this implies is just absolutely astronomical. So inside the cell we've got all of this transportation of the chemistry all around the place. Up and down these microtubules we've got transportation going on, let alone it actually getting outside of the cells altogether and into, for example, the lymph or the blood system and being transported around the body, and all these different ways that we've actually got stuff going on. All of it takes energy, all of it takes activity, all of it takes decision making. So consciousness is involved in all of this active transport of chemistry around the body. The next area that we really have a big focus on inside TRIM is the enzyme proliferation. So we talked about this a little bit before, but there's been some dramatic changes over the last 100 years. So particularly with how our environment has actually shifted, and our food in particular has changed completely. And all of the toxicity that goes with our pollution, whether it be deliberate pollution or unintentional pollution, there have been some tremendous changes in this. All of this has come to have an impact on these different classes of enzymes, and I just want to point out to you that there's two general categories of enzymes which these environmental factors come in and have an effect on. And once again, these are a very, very strong focus for TRIM, and that is that some of them are stimulating activity and some of them are actually doing the complete reverse. How this is ordinarily done is by having an impact on the competitive nature of what these enzymes are doing, and or indeed absolutely inhibiting the ability for them to actually come in and partake of the chemistry. So the last focus is actually the basic chemistry. So when I talk about basic chemistry, I'm talking about the digestion of nutrients for a beginning, so the whole process around the actual digestion of nutrients. And this is happening even within a cell. So a nutrient will go into a cell and then it'll be further digested. So there's all the things that are going on in your tummy. There's a parallel process in a way actually occurring inside each cell. But the key factor is that we've actually got a nutritional or digestive process going on there. Next thing that we want to look at is the way in which these basic nutrients, which are going to be some aspect of proteins, that might be micronutrients as well, or they're going to be fat or sugar. This is basically what's going to be going on inside your body. Various vitamins as well. However, when we're talking about what's going on inside the cells, the way in which the huge range of sugars are being manufactured has a big influence from consciousness as well. So the consciousness will be involved in which particular sugars are actually being produced, and indeed which ones are being consumed. So we know, for example, that fructose, which is a type of sugar, very simple sugar, can have tremendously negative impact on some of the functioning within the body. However, it doesn't necessarily have to. So depending on what we're doing with this kind of work, there's an opportunity to moderate and mediate the negative effects, for example, of a high intake of that type of nutrient. However, what we're really looking at is the way in which the body is managing the conversion between these different types of sugars, for example, between the different kinds of sugars, the different kinds of fats, between all the different kinds of fats, and back to sugars again. So we've got all of this interchange going on. In certain circumstances, where you really are running short of energy locally, well then even proteins can be broken down into sugars as well. On the other side of that equation, of course, we're building up all the parts that make the cells, and for example, things like the cell walls are made up out of effectively fats, are very complicated, beautiful fat structures that actually go to make up the cell walls. And the list goes on. So we have things which are part protein, part fat, and that are making up bits and bobs and inside yourself. And indeed, there's aspects of sugars involved in the construction of all of these parts of the body as well. So you'll get something which is part protein, part sugar, part fat, for example. And all of this needs to be happening, and all of it can actually be moderated by these consciousness processes that are going on. Okay, so that's all I wanted to talk to you about in terms of the science aspect of the chemical basis for cellular functioning, and I hope you found it useful. If you do have any technical questions about particularly the implications for diet and how diet actually relates to these things that we're talking about, but equally obviously the psychological issues which are coming in and impacting on it as well. If you have any questions about that, do pop them into the Facebook group, and I look forward to seeing you on the next module. So thanks for watching.